So central dogma of biology is this, going from DNA to RNA to protein. So the genetic information is going to go from DNA in the nucleus to RNA. The RNA carries the message to outside where the proteins are made. So this is what we're going to look at today. And the process of going from DNA to RNA is transcription, and RNA to protein is translation. This word transcription means to transcribe. So if you think about a court reporter, they are transcribing everything that's being said in the court. So taking information that's spoken, typing it up into written form. So spoken word would be DNA and typing it up would be RNA. So taking information in DNA and changing it to RNA. So that's transcription. Translation also means, you know, what it means in English. So if somebody spoke Spanish and you don't understand Spanish, you have a translator. They're going to say a word in Spanish, and then the translator maybe tell you the word in your language or in English. So that's translation. So in this case, it's taking the information from RNA and translating it and taking all the instructions and making the protein. So this is just a summary slide. We will look at it in detail. So a gene is a sequence of a DNA that encodes for a specific protein, and there can be multiple genes on a chromosome. On this slide, it says that protein production occurs in two stages, transcription and translation. There are three stages. The very first step is to copy the information from DNA. That's replication. So that's cloning. So for some reason, your book decided not to add in this chapter, but in the next chapter. So we're going to cover DNA replication in next chapter, and transcription and translation in this chapter. But DNA replication basically means just to copy the word, copy the information, so to clone. So I'm going to give you an analogy of cooking for this whole process. So this is the analogy that's being used in your book as well. So this cookbook has the information to make brownies. And this is your grandmother's cookbook. And she never ever wants the cookbook to leave the house. She don't want you to take the cookbook with you if you want to make brownies. So let's say you or your friend wants to make brownies, but your grandma, she said the cookbook cannot leave the house. So what you do, you maybe take a picture and you can text it to your friend or yourself, whoever's making brownies. And then your friend is going to basically get all the ingredients, assemble all the ingredients that are needed, and follow the recipe, read the recipe, and make the brownies. So in this case, DNA has to stay inside the nucleus. It cannot leave the nucleus. So that's the cookbook. It has all the instructions to make the proteins. So you take a picture, that's transcription. You take a picture and you send a text message to your friend. That's RNA. RNA is the text message. And now the information is out of the house. The information is out. Now your friend assembles all the ingredients. In this case, ribosomal RNA is where the protein is being made. So all the ingredients that are needed are amino acids to make proteins. So those are brought to this ribosomal RNA, and then the protein is made. So that's a brownie here. Brownie is a protein, amino acids are all the ingredients that are needed. Does that make sense? Any questions on it? So we're gonna look at it in detail now. So this is just an example how the protein production takes place. So transcription produces an RNA molecule that is complementary to DNA. In translation, the information in RNA is used to make a protein. What is RNA? What does RNA stand for?
Ribonucleic acid.
Ribonucleic acid. Do you guys know any differences between DNA and RNA?
RNA has one strand. why our
RNA,
DNA has two strands, and DNA is responsible to be encoding on genetic materials. Why RNA is its job is just to process protein, like make protein, process protein, and protein.
yeah, so DNA is double-stranded, RNA is single-stranded. There are different types of RNA. Some of them are, make proteins, and some of them just carry information. So let's look at some differences um, between DNA and RNA. So George mentioned double-stranded. So DNA is double-stranded, and then RNA is single-stranded. So RNA is much simpler than DNA. And that's one of the reasons there's a theory that RNA came around first, and later there were some mutations and DNA came around. But because DNA is more stable, it stuck around. So in some viruses, you still find RNA strands. Some of them have DNA. The nucleotide bases we learned for DNA, there's adenine, guanine, cytosine, and thymine. One another difference between DNA and RNA is RNA doesn't have thymine. Instead, it has uracil. So since adenine binds with thymine, now adenine is going to bind with uracil. So that's big difference there, and we will be using this when we do the transcription. And one last one is that DNA stands for deoxyribose and RNA is just ribose. And the reason for that is DNA is missing this O here. So there's OH, it's missing this O, therefore it's deoxy. There's no oxygen or O. So deoxy, if there is O, so there's no deoxy, it's just ribose. So that's another difference. When you look here, DNA, when it's pairing with RNA, adenine is going to pair with uracil because there is no thymine. Thymine is going to still pair with adenine because RNA does have adenine. Cytosine with guanine and guanine with cytosine. So that's complementary base pairs. So this slide is just highlighting the differences. One is the base, uracil, it's single-stranded, and our DNA is double-stranded. And then there's the OH group there, and it's missing that O, cis deoxy. So let's look at different types of RNA. So RNA molecule have a very important role to make this protein here. So this protein here. So there's three types of RNA. We learned about this RNA. This RNA is basically carrying the message from DNA to outside. So this RNA is called messenger RNA. It's carrying the message from DNA to outside where the protein is going to be made. Then there is ribosomal RNA, known as rRNA. Ribosomal RNA is the site where the protein is being built. So ribosomal RNA is doing work. So some of RNA molecules are enzymes, and one of them is a ribosomal RNA. This messenger RNA is not enzyme because it's not doing any work. It's just carrying the information outside. This ribosomal RNA is doing work, therefore it's an enzyme. Then there is tRNA or transfer RNA. This tRNA is going to transfer corresponding amino acids that are needed on this ribosomal RNA. So we will look at it in a lot more detail. So these are three types of RNAs.